The large number of genetic designs that get employed during synthetic biology experiments often puts a burden on conventional record keeping. Once you hit your 500th part, that paper notebook just won't cut it anymore as a means of tracking your sequences and stocks. Data management platforms understand the data structures present in your work, such as your oligos, plasmids, or cell stocks, and store this data. Effectively, they are databases with user interfaces and a small amount of business logic. The Registry of Standard Biological Parts was started at MIT in 2003 based around Tom Knight's idea of a biobrick part. Each brick encodes a piece of information such as promoter, coding sequence, or terminator, and there was one standard assembly method for assembling these parts together, the biobrick standard assembly reaction. Each part has an entry within this database presented to the user as the Registry of Standard Biological Parts website. A sample of plasmid DNA encoding the part is housed in a centralized repository and distributed. The part data stored in the database can be used to specify composite parts as a list of smaller basic parts. This way, you can simply specify the composition of a larger device, and the software can generate graphical representations and the full-length sequence for you. The registry also provides wiki functionality for users to describe the functionality, usability, or other explanations of the part. Each year, this collection of samples is distributed to every institution that participates in the iGEM competition. In response, participants are expected to provide new samples to the registry to grow the effort each year. All the part sequences are made public through the registry. Today, the registry is run by Randy Redberg from a non-for-profit group that also runs the registry. Thus, the two efforts are often regarded as one and the same. However, there are also registry users outside of the iGEM program. The registry is generally regarded as the first foray into the space of formalization and computerization of genetic engineering. The registry was innovative and has been highly successful amongst younger engineers. However, the culture of the field today prevents development of expensive and slow research in a fully open setting. The cost of high-value genetic information is too high right now for biotechnologies to operate without full control of their data servers and security over the access to their intellectual property. Users also wanted more functionality and more compatibility with non-biobrick work. This encouraged many research to develop alternatives to the standard registry. JBay ICE, also referred to as the JBay Registry or just ICE, was developed by Tim Ham at the Joint Bioenergy Institute, which is the biofuel effort run by Jake Hiesling. Instead of just one centralized database, ICE is open source such that many distinct databases can be set up. Primarily, one uses ICE to store, visualize, and share annotated sequence information. However, it also provides tools for automating the analysis of sequencing data and hooks into several design tools. ICE is the first BioCAD tool with the Web of Registries concept, in which a network of registries speak to one another, sharing genetic information. Clotho is an effort to connect together BioCAD tools into a coherent ecosystem. It houses shareable apps that perform distinct types of work on data objects such as samples, plates, and the like. Clotho was started by Douglas Densmore and myself, starting in the iGEM program for the first 2 years, 2008 and 2009. Shown here is a plate editing tool from the Clotho 2.0 distribution in 2010. The 3.0 version of Clotho will be released in 2014, and it will be the first enterprise quality version of the tool. It features dynamic schemas to represent a broader array of data concepts, and it also has web interfaces. Primarily, Clotho is intended as the glue to deal with the fact that software tools in genetic engineering typically are unable to interact with one another. It can also handle tools written in different languages on slightly different data structures and other such integration issues. GenoCAD is a mixture of computer-aided design, like it helps you design your constructs, and data management. It was developed by Jean Picard, and it is deeply rooted in the notion of standardized parts and part grammars. All parts belong to one part of speech, according to the central dogma. Because it wouldn't make sense, for example, to place a promoter downstream of its gene, this type of constraint on composition can be used to direct a user to designs that can work. It also enables an innovative interface in which you use a pull-down menu to choose the parts that might be appropriate for a given position in the design. These designs can be saved to your account on a centralized server.